Hello everyone, and welcome to this UCL Lunch Hour Lecture, which today is entitled "What Can Thousands of Brains Teach Us About Disease?" So my name is Professor Hélène Plain-Favreau. I'm a professor of cellular neurodegeneration at the UCL Queen Square Institute of Neurology. But before I introduce our speaker, I would like to start by mentioning that this year is a special year for UCL as we are celebrating our 200th anniversary. And to mark the celebration, we are just going to share a brief video with you.
200 years ago, there was no university in London, and we're really marking the coming of a very radical, very different institution to a city that was the largest city in the world.
UCL was to be open to people of all faiths, or indeed no faith.
Women were admitted to the Slade School of Fine Art when it opened at the start of the 1870s, and from 1878 women were able to come to UCL, study alongside men and get a degree, and this was way before any other university was giving degrees to women.
In UCL's bicentennial year, you can expect a major new exhibition, 3 books, installations from renowned artists, and UCL Illuminated, a spectacular immersive sound and light show on our central London campus.
The main chord is going to turn into a multi-day theatre with filming, workshop, lights, laughter, comedy, theatre and drama. It's going to be magic.
I'm really excited about telling people the story of how we as an institution have evolved over 200 years and how we're going to evolve for the next 200 years.
Thank you. So it is really now my honor and pleasure to introduce today's speaker, so Dr. Zane Jaunmuktane. And Zane really is an academic neuropathologist. She's based at the Division of Neuropathology and the Queen Square Brain Bank at UCL, where she's actively involved in advancing both clinical practice and research in neuropathology. So our areas of expertise encompass both molecular neuro-oncology as well as the analysis of peripheral nerve biopsies. Also, Zane specializes in neurodegenerative diseases associated with misfolded protein aggregation, with a particular focus on their mechanisms of progression and pathological diversity. So before we begin, I wanted to let you know that we will have some time at the end. The lecture is going to be about 40 minutes. We'll have about 20 minutes of a question. And this question, you'll be able to submit them at any point during the talk by going to Slido and entering the code #brains when asking your questions. So you should have the link as well in the invitation. And with that, I would really like to welcome Zane.
Good afternoon, everyone. It's a great honor for me to give today's UCL Lunch Hour Lecture during Brain Awareness Week on a research topic that I'm deeply passionate about. If we want to understand how to treat diseases of the brain, there is one unavoidable truth. We first need to understand what is actually happening inside the brain itself. For many neurological diseases, the most direct way to do that is by studying the brain after death. Over the past decades, these studies have transformed our understanding of conditions such as Alzheimer's, Parkinson's disease, and many other neurological disorders. Today, I would like to take you behind the scenes of this work. I will show you how studying the brain at postmortem has already helped us make important discoveries and how new technologies, particularly artificial intelligence, are opening entirely new possibilities for understanding brain diseases. In many ways, studying the brain after death allows us to see the final stage of the disease in that individual. But the real purpose of this work is not simply to understand what happened to that one person. It is to improve diagnosis and treatment for the patients who will develop these diseases in the future. As mentioned earlier, I work as an academic neuropathologist between University College London and the National Hospital for Neurology and Neurosurgery here in Queen Square. Neuropathology is the medical specialty that studies and diagnoses diseases of the brain, spinal cord, and peripheral nerves. Becoming a neuropathologist involves quite a long training pathway. It begins with medical school. followed by the foundation years, where doctors gain broad clinical experience across different specialties. After that comes 2 years of general histopathology training, learning how to diagnose disease by examining tissues under the microscope. This is followed by around 4 additional years of specialist neuropathology training, focusing entirely on diseases of the nervous system and muscle. So by the time someone becomes a neuropathologist, they have usually spent around a decade learning how to recognize disease in the brain. Most neuropathologists then work as NHS consultants, diagnosing disease in patients, while some of us combine clinical work with research, which is the path I have chosen. Neuropathology is primarily a diagnostic specialty. In most cases, we examine small tissue samples taken during surgery to diagnose diseases in living patients, for example, brain tumors, infections, or inflammatory conditions. But there is another important part of our work, that is examining the brain after death, particularly in patients who have lived with neurodegenerative diseases. And this work is extremely important because for many neurodegenerative diseases, the most definitive diagnosis can still only be made by examining brain tissue directly. Understanding neurodegenerative diseases is becoming increasingly urgent. Broad clinical conditions such as dementia and movement disorders represent one of the major medical and societal challenges of our time. The greatest risk factor for most of these diseases is age. As people live longer worldwide, the number of affected individuals is increasing rapidly. Within the next 25 years, it is estimated that around 139 million people may be living with dementia and more than 130 million with various movement disorders. So the scale of the challenge is enormous. One of the major difficulties is that these diseases are often very difficult to diagnose accurately during life. For some diseases, we now have some useful biomarkers, tests in blood or spinal fluid. But for many conditions, reliable diagnostic tools remain limited. At the same time, disease-modifying treatments are still very scarce. So understanding the biological mechanisms behind these diseases remains a major scientific priority. Before showing you what a brain examination looks like, I want to pause for a moment and acknowledge something very important. Every brain we study begins with a decision made by a patient and their family. These are individuals who have often lived with difficult neurological diseases for many years and who choose to donate their brain after death so that future generations might benefit from a better understanding of these conditions. It is an extraordinary act of generosity. And it is thanks to these donations that the discoveries we have made today have been possible. After brain donation, the first step for the neuropathologist is macroscopic examination, that is, examining the brain with the naked eye. Different diseases affect different brain regions, and sometimes we can see visible changes such as shrinkage in particular areas or enlargement of the fluid-filled spaces called ventricles, as shown in the right far image. These observations provide the first clues about the underlying disease process. After this, small tissue samples are taken and processed for microscopic examination. The tissue is embedded in paraffin wax and then cut into thin sections using a specialized cutting instrument called microtome. These sections are only a few micrometers thick. They are placed on glass slides through a water bath and then stained with various dyes or antibodies against proteins of interest. Traditionally, we examine these slides under the microscope, but increasingly, these glass slides are digitized and then analyzed on a computer screen. One reason these diseases are so challenging is that the brain is extraordinarily complex. The human brain contains billions of cells, and each of those cells can form thousands of connections. If you imagine trying to understand a city by analyzing every road, every building, every interaction between people, that begins to give a sense of the complexity we are dealing with. This complexity also explains why different patients may experience very different symptoms. Some patients may present primarily with memory problems, others with language difficulties or changes in behavior, and others with a range of movement disorders affecting how they walk, speak, or control their muscles. In many neurodegenerative diseases, the underlying problem involves proteins that misfold. Proteins normally have precise shapes that allow them to function correctly. But sometimes these shapes change and the proteins begin to accumulate abnormally. Depending on which protein misfolds, it will accumulate either in neurons, such as alpha-synuclein, tau, and TDP-43 proteins, in extracellular space, such as amyloid beta and prion, or also in glial cells, such as tau and alpha-synuclein proteins. And based on the misfolded shape and involved cell types, different diseases will develop. For example, if it is prion protein, prion disease will develop. If amyloid beta together with tau, it is Alzheimer's. If clinically the patient has frontotemporal dementia, it can be either tau or TDP-43 protein. If it's motor neuron disease, then most commonly it is TDP-43 protein. If Parkinson's, then it is alpha-synuclein in a specific shape referred to as Lewy bodies. And if it is an atypical Parkinsonism, then it is either alpha-synuclein in a different shape from Lewy body or it is tau protein. The situation becomes even more complex because some proteins can misfold in multiple ways, producing different diseases. For example, proteins such as tau or TDP-43 can take several different shapes and cause a range of different diseases with different clinical symptoms. Only for some, we have reliable clinical tests currently available, such as for prion disease, for Alzheimer's, and very recently emerging tests also for synucleinopathies. But for many other of neurodegenerative diseases, we still do not have reliable diagnostic tests available during life. Another striking feature of these diseases is heterogeneity. Two patients with the same diagnosis may develop different symptoms, and the symptoms can start at different ages, progress at different speeds, and show different patterns of brain involvement. Understanding why this happens is one of the major goals of current research. We know that genetics and local environment plays a significant role, resulting in different pathology distribution and clinical symptoms. But the full range of underlying reasons we yet need to discover. The biological diversity between different diseases and the variability we see even within the same disease is one of the reasons why diagnosing neurodegenerative disorders early remains so challenging. Postmortem studies have also revealed something very important. In aging brains, it is very common to find more than one disease process occurring at the same time. For example, someone with Parkinson's disease, which is characterized by Lewy pathology, may also have Alzheimer's type pathology or vascular changes. When researchers first began systematically examining brains after death, this finding surprised many people, because in medicine we often think in terms of one disease causing one set of symptoms, but the brain often does not follow that rule. In reality, several disease processes may be unfolding at the same time. That makes the puzzle much harder to solve, but also much more interesting scientifically. We know that many of the pathologies are relevant, but not all of them are relevant. But we are yet to discover which combinations and extent of pathologies are those which are truly clinically relevant and which are innocent bystanders. These additional pathologies may help explain why patients with the same diagnosis can experience very different disease course and also develop different clinical symptoms, sometimes including atypical presentations. But they also make diagnosis more challenging. For example, when we detect a biomarker that indicates a particular disease, whether in cerebrospinal fluid or blood tests or on brain imaging, we need to understand how to interpret that signal in the context of other possible disease processes that may be present in the brain at the same time. This complexity can also influence treatment decisions because different coexisting pathologies may affect how patients respond to therapies. Definite diagnosis, therefore, often still is only possible at postmortem examination, and the mechanisms of disease progression and reasons behind variability in symptoms and range of pathologies are still not very clear. Importance of postmortem research differs slightly from that in living patients. With diagnosing a disease in living patients, we aim to not only put a label on a certain pathology, but the main goal is to predict biological behavior of a given disease, provide indication of expected outcome so that clinical teams can decide on most appropriate clinical management and choose most relevant disease-modifying treatments. The central goals of studying disease in deceased patients are similar to those in clinical medicine, but with an even broader aim, which is to improve diagnosis and treatment for future patients. One important objective is to understand why patients with the same disease can develop very different clinical symptoms. This variability, which we often refer to as phenotypic diversity, also includes differences in how quickly the disease progresses. Another key aim is to identify biomarkers, that is, measurable signals in blood, spinal fluid, or on imaging or other tissues that could allow us to diagnose disease early in living patients. But it is equally important that we understand how to interpret these biomarkers correctly, particularly because many patients have more than one disease process occurring in the brain at the same time. And ultimately, the goal of this work is to help develop more effective treatments and to better understand how different patients respond to those treatments, including potential side effects. To answer these questions related to disease mechanisms and biomarker and therapeutic target identification, researchers need access to one very important resource, that is, human brain tissue. And already several decades ago, the need for material provision to global research community was identified. This has led to the creation of brain banks, where through patient involvement, donated tissue is carefully curated, characterized, stored, and made available for research. In the UK, there are 10 specialized brain banks in neurodegenerative diseases, curating cases with diagnosis of whole spectrum of dementias, movement disorders, traumatic brain injury, and importantly also controls, which are as important to study for comparison along with disease conditions. These brain banks are coordinated through UK Brain Bank Network. There are also additional brain collections and brain banks focusing on other neurological diseases such as brain tumors and epilepsy. Worldwide, there are, of course, many more brain banks specializing in a wide range of neurological diseases. Modern brain banking is no longer limited to collecting brain tissue alone. Increasingly, it also includes other parts of the nervous system, samples from peripheral organs, and importantly, samples of skin. These skin samples are particularly valuable because they allow us to study living human cells in ways that were not possible before. From a small skin sample, we can grow cells in the laboratory called fibroblasts. These are ordinary cells that normally help support the structure of our skin. What is remarkable is that scientists can now reprogram these fibroblasts back into a very early flexible state known as induced pluripotent stem cells, or iPSCs. In simple terms, this process turns back the clock on the cell, making it to behave like a stem cell that has the potential to become many different types of cells in the body. From these iPSCs, we can then guide the cells to develop into brain cells, such as neurons or supporting glial cells. This gives us a unique opportunity to study human brain cells derived directly from patients, but in a controlled laboratory setting. Even more impressively, scientists can grow these cells into small three-dimensional structures known as organoids. These are also sometimes described as mini brains. While they, of course, are not actual brains, they can mimic certain aspects of brain organization and function. This allows researchers to observe how brain cells interact, how disease processes begin and develop, and how different treatments might affect these processes. One of the previous lectures in this series, delivered by molecular neuroscientist Professor Selina Wright, explains this area in much more detail. Her work highlights how organoids are helping us to better understand the mechanisms underlying dementia, and I would strongly recommend listening to her lecture to gain a deeper insight into this exciting and rapidly advancing field. Using tissue made available through the generous donation of patients and their families, we can study brain disease across multiple biological scales. At one level, we analyze the clinical symptoms that patients experience during life. At another level, we examine the distribution of pathology across different brain regions in the brain itself. And at even finer levels, we study what is happening inside individual cells, analyzing genes, proteins, other molecules, and structure of the pathological inclusions. Researchers often refer to this type of work as multi-omic analysis. This means studying several layers of biological information at the same time. For example, genomics, which studies genes, proteomics, which studies proteins, and other molecular measurements that help us understand how cells function. In neurodegenerative diseases, this is particularly important because many of these disorders are driven by proteins that misfold and accumulate abnormally in the brain. By studying the structure of these misfolded proteins, together with genetic and molecular information and the pathology we see in brain tissue, we can begin to understand how disease develops and spreads through the brain. In this way, a multi-scale and multi-omic approach allows us to link molecular changes inside cells with the changes we observe in brain tissue and ultimately with the symptoms experienced by patients. Traditionally, much of our work has relied on careful manual examination. We study clinical records to understand symptoms during life, and we examine brain tissue under the microscope using special stains that highlight different proteins. This approach has been incredibly important and has taught us a great deal about diseases like Alzheimer's. But it is also time-consuming and depends on visual interpretation, which can vary between observers. In Alzheimer's disease, one of the key changes in the brain is the buildup of a peptide called amyloid beta. This protein gradually accumulates in a very predictable pattern across the brain. It typically begins in the outer part of the brain, known as the cortex, which is responsible for thinking and memory. From there, it spreads deeper into regions involved in memory, such as the hippocampus. As the disease progresses, amyloid beta continues to move into other areas, including structures involved in movement. Then it spreads into the brainstem. And finally, even into the cerebellum, which helps coordinate balance and movement. We describe this type of stepwise spread in stages known as tau phases, ranging from early to advanced disease. A second important protein in Alzheimer's disease is tau. Tau behaves quite differently. Instead of starting in the outer brain, it begins quite early in a very small, deep region close to the hippocampus, an area crucial for forming memories. From there, it spreads into hippocampus itself, affecting memory more directly. It then moves into nearby limbic region, which is involved in emotion and in behavior. As the disease advances further, tau spreads widely across the cortex, the outer brain, affecting many different functions. In the late stages, it reaches area responsible for specific abilities such as vision, hearing, and movement. Early stages of Alzheimer's disease are actually quite common in the aging population, and the way the disease progresses does not actually always follow the patterns I have just shown. This variation highlights just how complex the brain is and how complex these diseases are. This is where artificial intelligence becomes truly transformative. Most of us already interact with AI every day. When digital maps suggest us the fastest route, when streaming services recommend films or music based on our preferences, or when our phones recognize our face or fingerprint to unlock the device. Artificial intelligence is beginning to transform how we study brain disease, including neurodegenerative conditions such as dementias and movement disorders. In brain research, we now generate enormous amounts of information, including clinical histories, high-resolution pathology images, genetic data, and many types of molecular measurements. Traditionally, these different types of data are analyzed separately, often by different researchers using different methods. But increasingly, we are able to analyze them together. Artificial intelligence allows computers to analyze these very large and complex data sets and identify patterns that would be extremely difficult for humans to detect on their own. For example, AI systems can analyze clinical records, pathology images to either quantify or recognize inclusions, genetics, and molecular data simultaneously, allowing us to begin linking changes we see in brain tissue with clinical symptoms and underlying biological mechanisms. At the Queen Square Brain Bank, we have recently been fortunate to receive support from the Rita Lila Weston Trust to develop new ways of analyzing brain disease using artificial intelligence. The goal is to use these approaches to better understand how brain diseases develop, how they spread through the brain, and ultimately how we might diagnose and treat them earlier and more effectively. There are different types of artificial intelligence models that can be used for this. Traditionally, many approaches in medical research use statistical or machine learning models designed to analyze a single type of data. For example, one model might analyze written information such as clinical records, while another might analyze images such as pathology slides. These models can be very powerful for the specific tasks they were designed for. More recently, we have begun using approaches based on deep learning. Deep learning uses computer systems loosely inspired by networks of neurons in the brain. By analyzing very large numbers of examples, these systems can learn to recognize complex patterns in data. One advantage of deep learning is that it allows us to build multimodal models. That is, models that can analyze different types of data at the same time. For example, a model might analyze images of brain tissue together with clinical text or genetic information, allowing us to study relationships between them. But generally, they are usually task-specific, meaning they cannot easily combine different kinds of information. Even more powerful are what are known as foundation models. These are very large AI systems trained on enormous datasets containing many different types of information. Because of this broad training, they can learn more general patterns and can often apply their knowledge to new problems that they were not specifically trained for. In simple terms, these models allow us to begin linking what we see under the microscope with what we know about the patient from clinical records and molecular biology. At the Queen Square Brain Bank, we are now beginning to develop these kinds of approaches by analyzing digital pathology images together with clinical data and molecular datasets, allowing us to explore disease mechanisms in much more integrated ways than was previously possible. In neuropathology, we increasingly work with digitized pathology slides. Instead of examining tissue only through a microscope, the slides can now be scanned using specialized scanners that create high-resolution digital images of the tissue. These images contain an enormous amount of detail, often consisting of millions or even billions of pixels, allowing us to view brain tissue on a computer at different magnifications, much like using an online map where you can zoom in and out. This has several important advantages. First, it allows pathologists and researchers to examine the slides easily and repeatedly without needing to access the physical glass slide each time. Second, these digital slides can be shared with colleagues and collaborators around the world, which is particularly valuable for getting another opinion on a disease, for research purposes, and for teaching. And third, once these tissues are in digital form, we can apply computational methods and artificial intelligence to analyze the images. In other words, the slide becomes not only something that we look at, but something that we can also measure and analyze. This opens up entirely new possibilities for studying patterns of disease within the brain. One of the simplest but most powerful things artificial intelligence can do is to quantify pathology. Traditionally, when pathologists examine tissue under the microscope, we often describe the pathology as mild, moderate, or severe. However, computational analysis allows us to measure these changes much more precisely. When a pathology slide is digitized, it becomes a very large image made up of millions of tiny pixels, each containing information about color and intensity. If a stain marks abnormal proteins with a particular color, as in this case brown, the computer can analyze the size of those pixels and measure the color intensity within the pixels. This allows us to calculate exactly what proportion of the tissue contains the abnormal protein. But AI can go further than that. Instead of just measuring colored pixels and their intensities, the computer can also identify individual objects in the tissue, for example, protein aggregates or cells. It can then measure their number, size, shape, staining intensity, and spatial distribution across the brain. This allows us to build a much more detailed and objective map of how disease is distributed within the brain and allows us to analyze pathology in a much more systematic and large-scale way than would be possible by manual examination alone. Artificial intelligence can also be trained to recognize specific pathological features in brain tissue. To do this, we first show the computer large numbers of examples that have been carefully annotated and selected by expert pathologists. Through this process, the system learns to recognize patterns associated with disease. For example, abnormal protein deposits inside cells, aggregates between cells, or characteristic changes in the tissue structure. Once trained, the model can analyze new pathology slides and automatically identify these features. In some cases, these systems can even detect very subtle patterns that may be difficult for the human eye to recognize consistently. But the real power comes when we combine all these different types of information. By integrating pathology images with different cellular patterns and quantified pathology data with clinical data, genetics, and molecular biology data, we can build models that analyze disease in a much more comprehensive way. This type of approach is often called multimodal data integration. As a comparison, we might have individual satellite images, traffic maps, weather patterns. Each of these gives us useful information, but none of them alone tells the full story. Artificial intelligence allows us to combine these layers of information and detect patterns across them. And that is exactly what we are beginning to do with brain disease. When we bring all of this information together, new possibilities begin to emerge. For example, we can begin to develop models that can help us predict disease, estimate how pathology progresses through the brain, forecast clinical outcomes, and identify new biomarkers, and potentially identify new drug targets. In other words, we move from simply describing disease to predicting and understanding its behavior. To give a simple hypothetical example, imagine we are trying to predict the risk of dementia in a patient with Parkinson's disease. For that patient, we may have many different types of information: clinical history, genetic background, blood biomarkers, measurements of brain pathology, and increasingly detailed molecular information from brain tissue. Each of these on its own tells us only part of the story, but when we combine them together using artificial intelligence, we can start to identify patterns that would otherwise be very difficult to detect. By integrating all these layers of information, the model can begin to estimate when dementia might develop, identify what biological processes may be driving the disease, and potentially help guide which treatments might be most beneficial and when they should be given. So the goal is to move from simply describing disease to predicting its course and tailoring treatment more precisely to the individual patient. So far, I have mainly shown you how we study and can apply AI to research brain disease today, but science never stands still. Many of the tools we are using now did not exist 10 or 15 years ago. And some of the technologies that may transform this field in the future are only just beginning to emerge. Another important direction for the future of brain banking is improving the quality of the human brain tissue that we study. Traditionally, brain banks have focused on careful tissue collection and curation using standardized protocols to preserve the biological integrity of the tissue. This remains absolutely essential. But increasingly, we are recognizing that progress will also depend on engineering innovation, developing new preservation technologies, imaging systems, and instruments that allow us to study tissue in ways that were not previously possible. When these two areas come together, careful brain banking and technological innovation, we can begin to generate very high fidelity human brain tissue resources. And that opens entirely new possibilities for research. It allows us to study disease at much finer levels, including subcellular changes within individual cells, functional changes in neural circuits, how different regions of the brain connect with each other, and how disease processes are organized across space within the brain. In other words, improving tissue preservation will allow us to ask new kinds of biological questions about human brain disease that were previously out of reach. Last but not least, accelerating discovery will depend on international collaboration. At the Queen Square Brain Bank, we are contributing to these efforts by helping to centralize data repositories and digital slide collections, supporting the development of shared global research infrastructures. Before finishing, I would like to return to where this work truly begins. Every discovery we make starts with the generosity of patients and families who choose to donate tissue for research. Their decision allows scientists to study diseases that affect millions of people. This work is also made possible through the support of research funders and charitable organizations who invest in understanding how brain disease is developing and identifying better treatments. So when we talk about brain research, we are not only studying disease, we are also trying to answer one of the most fundamental questions in biology: how the most complex organ in the human body actually works. Because ultimately, to cure diseases of the brain, we must first understand what happens inside the brain itself. Thank you very much for your attention. I would be very happy to take any questions which can be shared through this QR code.
Thank you so much, Zane. It was really inspiring and really interesting lecture. So, as Zane just said, please don't hesitate if you would like to put a question in Slido. You have the QR code, but also you receive the link in the Eventbrite invitation, or you just go to Slido and you enter the code #brains. I think it might take a little bit, but I see there is already a question for you, Zane, in Slido. The thing is that it was posted 25 minutes ago. I tried to look when it was, so I might ask you, Milan, thank you for your question, if you can tell us, because the question is, how are these tests conducted? But because, we don't know exactly when the question was posted, I think it was for the omics, but Milan, please tell me if I'm wrong. And how are the omics conducted, and can they be done while the patients are still alive?
Yeah, so examining the neurodegenerative diseases in living patients on tissues is not an easy task, because the neurodegenerative diseases develop over a long period of time, and there are not many areas in the brain which we can easily sample without causing any functional defect in the patient. So therefore, unfortunately, studying the tissues with regards to neurodegenerative diseases in living patients is not really a feasible possibility. In terms of how we carry out multi-omics studies, so all the donations are prepared in a way that there is tissue available in a fresh frozen state. And we take small samples from these frozen brains from areas of interest, and we prepare them depending on the multi-omic study for the downstream analysis. But yes, all multi-omic studies, or majority of them, are done from these small frozen samples.
Thank you, Zane. I think it was for the multi-omics, but as I said, Milan, just please say if I when I've seen the question. So while you're posting your questions, consider them at the moment. Maybe I'm going to use my privilege of chair to ask one, Zane. And you mentioned quite a bit AI, obviously, and also at some point how AI could make a much more precise, in a way, kind of analysis of the brain. I wanted to know, in your experience, where do the human experience, or maybe intuition, I don't know, still overpower actually the AI in interpreting the brain pathology?
Yeah, that's obviously is a very important question. I do generally think that what artificial intelligence and data-driven modeling enables us is to really just skyrocket the human interpretation of the neurodegenerative disease. The human interpretation and the human knowledge about the diseases has never been as important as it is right now in the era of artificial intelligence, because it's only by putting things in the context with multi-layered information we have acquired over the years through studies, only that's when the artificial intelligence can really provide us a meaningful, valuable information, which will be useful for new mechanism identification, new treatment identification, new biomarker identification. So yeah, I'm a strong believer that artificial intelligence is helping to advance our interpretation of the results, but it's not really helping us to do the experiments for us.
Okay. There is a question from Rita. Thank you, Rita, who first of all is thanking you, Zane, for an amazing presentation and asking what do you find to be the most challenging part of working with the brain at the moment?
That's a very good question. I think the most challenging parts most definitely would be to do with the frustration that it is just so complex that we have made so many discoveries already, but yet we cannot really cure the disease as yet. And because it's all to do with the complexity, and that obviously for people like me working with the material on day-to-day basis is also frustrating that it's just so very complex and there are just so many factors we are just even now discovering which are contributing to the complexity. So yeah, I think that the challenging part would be the complexity of the disease.
I understand. Yeah. As it is frustrating but fascinating, I suppose, at the same time, right? Tanya has a question for you as well and ask, how does AI performance compare to clinicians in early stage dementia detection, especially MCI?
Yeah, so MCI stands for mild cognitive impairments, and definitely so artificial intelligence is taking and helping across the domains, not only on interpreting the tissue-based data, but also the clinical data. At this moment in time, I'm not a clinician, so I obviously cannot give the full details. I don't think at this moment in time one can rely fully on artificial intelligence of interpreting mild cognitive impairments, but it definitely, as across the domains, across the medicine, it definitely, I am absolutely confident that the time will come when artificial intelligence, data-driven modeling-based approaches will also massively help make more accurate separations between MCI and different stages of dementia. But we are not there yet, no.
Tanya has another question on AI, but just before I would like to thank Milan because actually, yes, I probably picked up his question a little bit late and he thanks you for the genomic answer and it's a fascinating lecture. He said he really enjoyed it, but he was referring to the quick, sorry, when he was mentioning DSA and not the genomics in my first question. I think when they were asking whether DSA could be performed with in-life patients.
Yeah, so that is absolutely true, and that's exactly how prion diseases, Alzheimer's disease, recently also synucleinopathies, how they are diagnosed in living patients. So it's not on the tissues, so not on the brain biopsy, but on the cerebrospinal fluids, which is taken from living patients, and in the blood there is also a very strong hope that in the next few years there will be a very good plasma marker to diagnose Alzheimer's disease in living patients using the plasma samples.
Thank you, Milan, and apologies for I had picked up your question a little bit late. Tanya has another question, and she's asking what level of explainability is needed for clinicians to trust AI outputs?
Yeah, so that's a very, very important question, which also links to some of the previous questions, and my answers to that is that it really goes back to the knowledge that that it could not be more important. It couldn't be more important at this moment in time to have that background knowledge to be able to interpret the artificial intelligence provided results. That it all goes back to that the knowledge will be as important as it's ever been for us to understand how much we can rely on the result and how much the result is true and not a made up hallucination.
Emily has another question for you. What originally made you want to go into neuropathology and what keeps you motivated? That's a very good question.
Yeah, thank you. Obviously, that's also a little bit personal question. So when I was 10 years old, I had meningitis. And that's when I spent a lot of time in hospital, and I got hooked up by neurology specialty, by medicine. And that's how I went into medicine. And then when I was the first year medical student, I joined a research project. And my involvement in that research project, which was in neuro-oncology, was in neuropathology. And I never found anything as interesting as neuropathology when I rotated through the medical specialties, through the medical training. And yeah, so from the first year of medical school, I knew that I'm going to be a neuropathologist. And what keeps me motivated is really, as already said before, was it's frustrating that it's so that the brain disease is so complex, and there are just so many layers why we cannot find a cure for the disease. From the scientific point of view, it is also very, very interesting that complexity and that multi-layer element which is there, that just makes it so interesting and motivating to study. And yes, I work part-time for NHS and in Queen Square Brain Bank with my research in neurodegeneration. I think also that range of through neuro-oncology, through nerve biopsy interpretation, through diagnosing neurodegenerative diseases is also what makes it a very, very interesting job.
Thank you, Zane, and thank you, Emily, also because I know Zane a little bit, but I didn't know that about you, so that's another privilege of sharing the lecture. Maybe I have another quick question for you. We still have 5 minutes, so and please, guys, if you have other questions, don't hesitate to put them in Slido. But you mentioned the organoids or the mini brains, and you said yes, but they're not totally brains. What is missing, you think, in the for us, if you're more cell biologist, in the organoids or the mini brains in a petri dish, as we could say, that is not there, if I can say that.
Yeah, so I guess the soul is missing, the emotions are missing. But yeah, so what is there is that is an absolutely incredible laboratory model to study the disease in a living condition, and the ability also at Queen Square Brain Bank to have the post-mortem material and then derived organoids from the same samples, and then to do that multimodal integration and correlation is definitely something which will accelerate the research discovery.
And from a pathological standpoint, do you think it might be very variable between one disease to the other, but how early do you think we can detect the first changes in the brain before the clinical symptoms appear?
So the thing is that if we have the brain available for one reason or the other, whether the patient died from another disease, we can see those changes in post-mortem material. What is still the challenge is to identify markers, either imaging markers or fluid markers, how we can translate those early changes we see in the brain tissue, how we can translate them and detect them in living patients, because the changes are so subtle. In the post-mortem material, it's not a problem for us to see it, but because the changes are so minimal, the more visible effects as on imaging haven't developed there yet, or the ones in the fluid markers. And it's just really improving the technology and understanding those subtle differences to identify them in living patients. That's the bridge we still need to cross.
So the imaging and the biomarkers, or for example, the atypic like melanoma.
Yeah, and I could not emphasize more that the technological advancement in these engineering innovation abilities is undeniably what also makes this a very, very exciting time.
Thank you so much, Zane, I think, and we are pretty much right on time, but I think if we have no more questions, maybe we could actually put the final slide here so that everybody can see. As we said, it's the UCL 200 2026. You will know that by now. And to know about our next lunch hour lecture, which will be about 50 years of becoming a mother. And this will be this Thursday, 19th of March, again from 1 to 2 p.m. So please join me in thanking Zane for a fantastic lecture, and all of the audience for your really interesting questions. Thank you again all, and have a lovely day. I hope you can all enjoy a little bit of sun today. Thank you, Zane.
Thank you.